So we have an exciting panel here. These are our pharmaceutical executives. I'm going to introduce them briefly to you and ask them to come up on the stage. Many of you, I think, already know Jay Bradner because he was with us for many, many years. He is now president of the Novartis Institute for BioMedical Research, and he's a member of the executive committee at Novartis. And he is familiar to you because he was a faculty member at Dana-Farber and a professor at Harvard Medical School in our Department of Medical Oncology. He has been very instrumental in developing new drugs while he was at Dana-Farber. He founded 5 companies, and he's brought all that expertise to Novartis. Our second panelist is Jeff Leiden, and Jeff and I have been friends and colleagues for, I think, 30 years now, Jeff. Jeff is an MD, PhD, who spent many years in academia. He's a cardiologist, was chair of cardiology at University of Chicago. We then recruited him to Harvard, where he stayed for a whole year before departing for Abbott, where he was a chief operating officer and also the chief scientific officer. From there, he went to Claris Ventures, and from thence to being the CEO in 2012 of Vertex. And as you know, Vertex has had an amazing record, and just in the last couple of years, in developing new drugs for patients with cystic fibrosis that really transforms their lives. And finally, but not least, we have David Schenkein, who is the CEO of Agios. David is a board-certified oncologist specializing in hematologic oncology. He still sees patients at Tufts, but at the same time, he is the CEO of Agios, a company founded several years ago by Lou Cantley, Craig Thompson, and Tack Mack. And again, much success. They have just gotten FDA approval for a new drug for acute myelogenous leukemia that targets proteins known as IDH1 and 2. So please come up on the stage, and we'll start our discussion about the relationship between pharma companies, biotech, and academia, and my own personal feeling that this is an absolutely critical relationship that is really a marriage made in heaven for our patients, the fastest way to get new discoveries from the bench into the bedside. Okay, well, I'm going to ask you a personal question first to each of you, and that is, what attracted you first to science and then to the private sector? How did that transition happen? Not just the transition, but what was your initial draw to being a scientist? Start with you, Jeff.
Sure. So I think, well, I'd be interested to hear what you guys say, but I have lots of friends who are scientists, including Laurie, and one of the remarkable things when I ask them that question is almost all of them can tell me about something that happened to them, not when they were in graduate school, not when they were in college, not even when they were in high school, but often when they were in grade school or middle school, that really turned them on to science. And for me, I had a 5th grade teacher named Mrs. Giancana, who was a science teacher. I remember her well still. And she would let me come after school and do extra science experiments, including things like dissecting frogs, which for a 5th grade boy is a really exciting thing. And she hooked me on science very seriously, and from that point on, that's all I really wanted to do. With respect to industry, that was much more of a gradual evolution, Laurie. My lab, like both of their labs, started off doing very basic science on the development of the immune system, the development of the heart. And we started to realize that some of the technologies that we were developing could be used for therapeutics, in particular gene therapy. And so we sort of stumbled into it. And we tried to do that in academia. We're going to talk about that, I know. We then founded a couple of small biotechs, and we tried to do it there. And ultimately, I sort of came to the conclusion, with the help of some of my mentors, that if I really wanted to take basic science and turn it into medicines, you had to do that in a larger organization where you had the resources to actually discover and develop drugs, which, as I'm sure we'll talk about, it's a very complex, very complicated thing that takes teams of hundreds or thousands working together for quite some time. So it was sort of a gradual evolution as I went from academia to biotech to big pharma and then to big biotech.
Well, it's wonderful to be back here at the visiting committee. I think this is my 10th visiting committee, but a first in this new capacity. I went to college to be an English major, to be a writer. I was really taken with American poetry and literature. But my high school chemistry teacher, Mrs. Mueller, in Chicago, made me promise that if I were to go to Harvard, you have to take chemistry. There are so many great chemists at Harvard. And so dutifully I did. And in the second semester of organic chemistry, I had a very charismatic professor, Stuart Schreiber, who really, for very much the first time, studying what's called molecular recognition, the way that molecules bind to their target proteins. Science, to me, emerged as a creative art, not just atoms and numbers and calculus and vectors, but there was something very beautiful and malleable and three-dimensional about what we called chemical biology in my academic lab, and we unapologetically called drug discovery at Novartis. And so I guess I got hooked on science in the classroom first, but then ultimately in the lab back in Chicago at Northwestern in the summer times with a very brash graduate student who I just thought was the coolest guy ever, Seem Ansari. You know, at the bleeding edge of knowledge, you would use these gifts of chemistry and physics and computers to make first observations about the natural world. So it was a puzzle until I got to medical school. And then on the wards at University of Chicago at that time, and then doubly so when I came here to train in cancer with all of these faculty, it was less abstract, like a puzzle, and more of a mission or a calling. The idea that only through atomic level understanding of the diseases that affects these patients can we really have an impact. And so I really got hooked on science through chemistry, but I'm in science because of cancer. The second answer to your question is a little easier, but bittersweet to talk about in this room especially. I was having the time of my life at the Dana-Farber, leading a chemical biology lab, seeing stem cell transplant patients and working with the best doctors and most inspiring scientists, I think, anywhere in the world. But we had just started to move our molecules into patients, which was a transition I hadn't expected that we'd be able to do in academia. But we did, and people were responding to these drugs. I had treated a lot of mice in my day, some zebrafish, even a couple frogs. But when my heroes, Dan D'Angelo and Jeff Shapiro, and they were ringing us up and showing us these patients of theirs in horrible circumstances responding to these medicines from our fume hoods, it really changed me. The idea that this science could really have an impact. And in the environment that we have at Dana-Farber, not unlike the environment at Novartis with 6,000 scientists and $9 billion devoted to make breakthrough drugs, I went to industry still in pursuit of historic impact on cancer and other serious diseases.
So, Laurie, thanks for the opportunity to be here today. Thrilled to be here. So, I grew up in New York City, and when it was time to go to high school, I was living in Queens. 3 of my friends and I decided to apply to Stuyvesant in Manhattan. We didn't even really realize it was a science and math school until we got there. And I really wasn't all that smart to go into math. But I had a chemistry professor the first year in high school.
Seems to be a common theme.
Yeah. Who was really extraordinary and just lit up the world for me. And so as an undergraduate, I went as an undergraduate as a chemistry major and decided to actually go to medical school because of my pediatrician, who was a really important role model for me. So that's what got me into medicine. And then into industry, I went into industry pretty late. And so I had been on the faculty taking care of patients and running a research lab for almost 14 years at Tufts in hematologic malignancies, and never thought in a billion years I would go into industry. Mm-hmm. And I think what happened, 2 things happened. One is I met somebody as a favor, and that was a gentleman by the name of Mark Levin, who was the CEO of Millennium at the time. And Millennium had just bought a company that had a really unusual drug in very early clinical trials that you may have heard of from Ken Anderson earlier today, called a proteasome inhibitor, which is now called Velcade, and they had no group to develop it. And I realized that in the clinic I was helping one patient at a time, but if I could actually bring a drug forward all the way, you'd help tens or hundreds of thousands of patients. And so that's what drew me to Millennium. And then from Millennium, Genentech, Agios.
But Laurie, it's such an important question because you heard the same answer. You always hear that answer, right? Which I think teaches us something, which is if we want to train and create more scientists and more physician scientists, we have to start early. And we're sitting in this sea of talent here in Boston, including a lot of underprivileged kids who don't have the chemistry teachers that you had or the biology teacher that I had. And so I think as we think about it, certainly at Vertex, focusing on STEM education and STEAM education for these young kids, developing them at that stage, that's how we're going to get the next generation. If we don't, we're actually giving up a huge opportunity for them and for us.
And I would add to that special attention to gender.
Definitely.
Because young girls
Definitely.
move away from science and math very early on in their careers, and we have to intervene not in high school.
Definitely.
We need to intervene in kindergarten. And it's wonderful work you're doing at Vertex with your education initiatives for underserved in medicine and science. And we are in danger of losing the next generation of scientists, not just because of this, but also because of lack of funding from the NIH.
Sure.
And, young scientists are in laboratories of wonderful mentors, and they see that their wonderful mentors who are doing great work aren't getting adequately funded. This doesn't happen very often at Dana-Farber, to be frank. Nevertheless, it's happening around the country. And they get discouraged, and they say, well, why do I want to take on science as a career when it's going to be so tough and it's going to be so hard to get funded? So I'm glad you brought that up, Jeff. So maybe we could turn to the question of the different ways in which pharmaceutical or biotech companies can relate to an academic medical institution, and what you think are the ideal ways to do that. What's the best structure for those kinds of partnerships? Because there are many different possibilities, and many of them have worked and some of them haven't. So why don't we start with you this time, David?
Yeah, so, all of us have lived in academic medicine and now have been on the industry side. And I think historically, at least my view, particularly here in Boston in the 80s and early 90s, there was this wall between academia and industry, and you were viewed almost as a second-class clinician or scientist if you transitioned into industry. And that's changed, and the quality of the science being done on both sides now are both focused on how do we make medicines that change people's lives, because that's what we're all here to do. And I think as that wall has come down, with important conflict of interest barriers and stuff like that, as those walls come down, I think it's improved our ability to make important medicines. And so from my perspective, the two obviously important areas, we're a small company, that we interact with academia, is on the research side. We want to be as open as possible, and we're pretty open, and so we try and establish as many scientific collaborations as we can, which are where the incentives are aligned. It's good for both of us. Again, if we keep that common goal of making a medicine for patients one day and understanding the biology. And then the second, obviously, critical area is around clinical trials. So this institution, Dana-Farber, is one of our go-to phase 1 sites for our new drugs. And it's critically important because the physicians here and at other major academic centers are the ones who are going to really know and get the feel for, is our drug helping patients? Is it effective? Is it safe? How should we use it? We can't do that within the walls of industry. That has to be done here. So I think if we can continue to establish the relationships, and it's not ideal yet, but if we can keep the walls coming down and really make it aligned incentives to make important medicines for patients, that'll move things along much faster.
I agree. I mean, if a lot is said about academia versus industry, I find it totally distracting. I was bracing for a real change when I got to Novartis, Swiss pharmaceutical company, suit and tie. Turns out suit, no tie. And look at these gentlemen.
You look like triplets.
The big surprise is just how similar it is. Same instruments. Actually, a lot of the same people when I saw Glimcher. Same types of targets, same types of challenges. same mission, orientation. And so though much is said about academia and industry, my personal experience is that they're way more similar than different. And we should celebrate this collaboration in the space of clinical investigation, as David said, because only through that engagement in early stage trials such as at Dana-Farber, in late stage trials as we work with the TIMI group at the Brigham and Cardiovascular, are truths about therapeutics learned. And it's so productive and doesn't need much optimization. It could stand to be less expensive. But in the early discovery moments, pharmaceutical companies tend to operate very, very privately, very, very secretly. And the truly great pharmaceutical companies of the future will need to be much more of an open framework. And I'm, as you must know, really passionate about open science. I remember when they contacted me for this role, I said, well, you do know I practice like an open science. They said, yeah, we need to adopt that strategy. They had felt they had missed checkpoint therapy and immuno-oncology because Novartis was too closed, too sheltered. And so we're working really hard to open the framework of our research. We have a wonderful program with the Dana-Farber. I think it's $11 million or more a year to discovery science, to find new targets, to make new measurements that guide therapeutics, going on perhaps its 7th year now. And the insights learned and the bonds and cultures shared are very powerful at prompting the next generation of therapeutics. We just had a medicine approved called CAR T-cell therapy. These are T-cells from patients. We stick a gene into the cell, we infuse it back into the patient, and over 80% of children with refractory leukemia have complete responses, and many of them alive even 5 years later. And this technology was innovated at the NCI and at the University of Pennsylvania and other academic centers, but only through an alliance with Penn focused on this paradigmatic therapy could it become a reality for patients everywhere. And so we think about these learnings because if we can make our best practices our standard practices, then all the programs will work well in this way. What I think about all the time is I never called Novartis when I was a professor at the Dana-Farber. Mm-hmm. Never even occurred to me. Now they'll steal my baby, or they won't value my insights, or will not see the value of our technologies. And my hope is that these institutes that are in all the hot spots of innovation around the country and the globe, really, they have hulking discovery capacities. And could they be available, DNA-encoded libraries for Nat Gray, or DUB profiling systems for cerebrolage, could they be available to investigators in a non-predatory way that really, like the rising tide, floats all boats? And we're working hard on this. And honestly, Dana-Farber is the pilot for much of this practice of open science.
Well, you wouldn't work at what you didn't call Novartis, but I was glad that you were helping out Agios when you were here.
I was. And actually, David's labs at Agios and our labs at Novartis are connected by a barricaded tunnel. So we could do better too.
It's true.
So Jay, I think the last thing you said resonated with me, and it resonated with me because I think you're exactly the kind of person to go lead the Novartis Institute who will make those changes. I think that the ability for you to understand both sides of the equation and to make the case that you just made to investigators around the world, not only the Dana-Farber, that's an important step forward. I think that's what David was talking about at the beginning, that the walls used to be up, even between academia and biotech, certainly, as you say, between academia and big pharma. The way they're going to come down is to have people moving back and forth across those highways who understand both sides, and that's why I think it's particularly exciting that you're at Novartis. We talk about this as sort of an ecosystem where different pieces play different roles, but they have to all work together. And I think it's a really important thing for those of you that aren't in science to sort of grasp. Because if you take this as the concept of an ecosystem, what it means is that if any individual part of the ecosystem gets damaged, the whole thing grinds to a halt. And so when Laurie talks about NIH funding, the beginning of the ecosystem is the academic labs that do so much of the basic biology and the basic human medicine that leads to these therapeutic discoveries that we're all talking about. And they do that with NIH funding. And they also do the training, frankly, of all of us and all the individuals who eventually carry all this out. If we damage that part of the ecosystem by inadvertently or advertently taking NIH funding down by 8 to 17%, as was proposed, we stop the whole thing from happening because we don't get the next generation of trainees, we don't get the next generation of basic science or medicine discoveries, and so all three of us have less to work on as it comes down the pike. The next part of the ecosystem is that biotech world where people from academia spin their ideas out into companies to begin development, companies like Agios, companies like Vertex 25 years ago, and those are funded by venture capitalists. And so if we inadvertently damage the venture capital system or inadvertently damage the biotech environment, we've made a wall or a cut between academia and the next step in the process. And then you go to larger companies now like a Vertex and Agios or a Novartis, and they develop these drugs in thousands of patients around the world to prove whether or not they really work. And of course, they get them to patients, the access issue. And so every part of that ecosystem has to work. And my fear as I look at things now is that if there's not an understanding of that, inadvertently political and other changes can come in and damage one part without realizing they're damaging the discovery engine that's led this to be, the last 50 years, I think the golden time in biomedical research, almost all of it coming out of the US, by the way, in terms of new therapies for cancer, for heart disease, for cystic fibrosis. And the next 20 years can be even brighter because the science has never been better. But if we put up those walls or inadvertently damage a piece of that ecosystem, starting with NIH funding in academia, it's going to be a shame because we'll give up that opportunity.
So let me pursue that theme for a little while because we are under threat, right? Academic medical centers, all academic medical centers, no matter how great they are under threat. Financial compression. Healthcare reimbursements, especially in Massachusetts, have declined. While medical costs rise at about 3% a year, our increase in healthcare reimbursements is about 1% to 2%. And so every academic medical center needs to figure out how it's going to stay alive. And one of the ways to do that is to monetize its discoveries. Mm-hmm. And there is the sense that academia gets the short shrift when it comes to monetizing discoveries that our scientists have made. So that was one of the reasons, and of course, Jay, you know this better than I do, for installing a chemical biology program at Dana-Farber, which has been very successful. I think our goal at Dana-Farber is to do first-in-human clinical trials of molecules that we have developed in-house. And that is not only because we think we can do it and get them to patients more quickly, perhaps, but also because it becomes an evergreen source of revenue for us. Because if you can get through a phase 1A and phase 1B clinical trial and show some efficacy, even minimal as it is, the value of your discovery, the value of that compound goes way, way up. And that then brings funds, very desperately needed resources, back to academia to fund basic research and more discoveries.
So I'm going to agree with you, but disagree with you.
I know you are. I was setting you up.
I'm going to violently agree with you to start with, because I usually agree with you, Laurie. By the way, I've known Laurie, I won't tell you how many years I've known Laurie, but she's pretty much always right, so we'll just start with that. So I totally agree that basic science discoveries and laboratories can be monetized, and it's an interesting approach. It is, if you look at the history across all of academia, the fact is that most universities actually lose money on pursuing their patent portfolios, so that's something to take into account. But the real key question is how to do it, right? So I'll tell you a success story that suggests one way that comes from our world that you know, which is the CF Foundation. So right. The Cystic Fibrosis Foundation, as you know, raises money from patients and families and friends of patients with cystic fibrosis in an amazing way. I mean, they've raised billions of dollars. They had a dilemma 25 years ago, which was, should they build their own research labs and develop their own molecules with the money they were raising? Because that was a possibility. Or should they actually use the money to fund either academic researchers or companies that were developing drugs? And for those of you who know, there was a huge debate actually within the CF Foundation about that. In the end, they decided to use their money to fund academic labs and companies that were developing drugs. And that turned out to be, first scientifically and medically, an incredibly successful approach because the gene was discovered on their watch, partly with their money. The drugs that Vertex made, they gave us a very early Small but very important grant very early on in the process to help us grow the cells from the patients and help us organize the trials. And I think that we would not have done this without that small grant. But they did it in a really clever way, which is they asked for nothing up front. They gave us the money to grow the cells and do the trials, or help us do the trials early. But they asked for a royalty out the back end if we developed anything. And it was not a horrible royalty, a modest single-digit type royalty. On the basis of that, as we've developed drugs, and their royalties have kicked in, you may know they just sold their royalty for $3.6 billion to Royalty Pharma. They are the best endowed foundation in the entire world. In fact, their problem now is what to do with that $3.6 billion, because the CF journey is in many ways coming to an end. We're going to cure that disease. But it's just an illustrative case, I think, where you have to ask yourself, is it better to take that kind of deal, which has risk, for your molecules, because it's back-end loaded, you don't get a lot up front, but when it works, you get a lot? Or is it better to spend a lot of resource trying to discover and develop drugs in an academic institution, which can be done, but I will tell you is very difficult to do. We've all seen that tried many, many, many times without success. So it's just an open question for you to think about. I don't know, Jay, what you think?
Yeah, I would agree, Laurie, that I think it's critically important, obviously, that academic institutions not only survive, but thrive. And so obviously getting the appropriate resources to do that, given what's happening at the federal level, is vital. And it's certainly, I think, not an unreasonable way to think about doing it. I think the only issue I would have is, will that distract from the overall mission of the academic institution in terms of doing more basic biology and obviously patient care? Because the research we generally do, and so far, we're about to put our 6th molecule in the clinic. Everything's been homegrown and developed in-house, but our work is much more translational. Obviously, in our first example, we'd actually discovered the basic biology behind the target, but then it's very translational. And so the question, and the ecosystem, as Jeff pointed out before, is so critical that there's basic biology being done, and then there's translational work, and the two are beginning married together. But I think, appropriate compensation should be, if the discovery really comes out of the academic lab, there needs to be a mechanism to make sure that that's monetized in a fair way. And if it's by waiting till after the first clinical trial, if it can be done, if you have the machinery to be able to do that, then I don't have an issue with it. It'll make it harder for small companies like us to compete for those products. They'll all go to Novartis that has deeper pockets. Small companies like us won't be able to compete for those, but so be it.
Well, I hate to be disagreeable, but I think there's a huge opportunity for centers like Dana-Farber to not just participate, but to lead in therapeutic discovery science. I think there's a huge opportunity. I think Dana-Farber already does. I believe that therapeutics is a science. It's not the application of basic science. It's surely guided by basic science, but science has become so interdisciplinary. The science of our labs at the Longwood Center, we're non-disciplinary. Organic chemists, next to cell and molecular biologists, next to computer scientists, next to doctor. Focusing on a really challenging problem, recognizing that the only relevant model system of disease is the patient with real disease. And there, at the Farber, already exists such capacity in target discovery, such capacity in early drug development. It's only natural that rather than sit around with your fingers crossed waiting for the cure to cancer, that you take arms, that you'd organize around that ambition, not around the commercial ambition, but around the scientific ambition, the science of therapeutics. Call it chemical biology, call it drug discovery, it goes by many names. But there is a lot to be learned about therapeutic likeness. There's a lot to be learned about the biology of cancer through the lens of a small molecule probe that becomes the prototype for true drug discovery. I mean it when I say Dana-Farber has quietly assembled the best chemical biology department in the world, the most translational chemical biology department in the world. And not by recruiting National Academy fancy scientists, but by growing this farm league of people thinking about science and medicine in a different way. So I think the key is to execute against the strategy opportunistically, to be sophisticated to capture the value created and to understand the radical value that exists in parts of the Institute like the Longwood Center, while not eroding the culture of scholarship. There's a way to do discovery science with high scholarship and published in top journals, and if you don't believe me, go to NACRA's website, to train people in a new type of science, while also capturing the putative value of these technologies. And I think, Barrett, through your work, the culture of scholarship remains firmly intact as we spun out our 5th and 6th biotech, as we sold 3 of them to Roche and Celgene and Medivir and took one of them public. We didn't get any less scholarly. But we, Ken and I used to joke that we'll be the biggest donors to Dana-Farber someday through the royalty
Write that
streams
down.
on these molecules.
Sue is recording that right now. I think one lesson I take from what you've all said is that this kind of discovery can only be done in very special places.
Yeah.
95% of academic medical centers are not going to be able to do what we've done at Dana-Farber. But I do think there are some, Dana-Farber included, and I think the Tri-Institutional Therapeutic Discovery Initiative that we put in place when I was dean at Cornell with Rockefeller and Memorial Sloan Kettering also is an example of that. But it is rare. I would grant you, Jeff, that it is a rare academic medical center that can do that. But pharma can be big and glacial,
For sure.
slow-moving, enwrapped in bureaucracy. One of the things about being in an academic setting where you've got basic scientists talking to translational scientists, talking to clinicians in a seamless way, is that you can circumvent some of that red tape.
For sure.
Let me just bring up one other topic, and that's the FDA and all the new breakthrough statuses, et cetera. So, there's been a lot of talk in Washington about how ineffective the FDA is and why do they take so long to approve drugs, et cetera, et cetera. From my perspective, I actually think the FDA has been doing a pretty damn good job in getting drugs through fairly quickly. We had a good FDA commissioner in Rob Califf, and I think that Scott Gottlieb is going to be also excellent, in part because he has experience in the private sector. But I'd like to ask you, how are you all working with the FDA in terms of further accelerating the speed with which drugs get to patients? And what can we be doing as in academia to help you with that?
So I agree with what you just said pretty much across the board, Laurie, this time. First of all, breakthrough therapy, we actually spent a lot of time working with the FDA to actually write that legislation. Mm-hmm. And so they definitely involved industry in trying to understand what they could do better. And it's had a major effect. We've all had that, I think. Are your drugs breakthrough as well? We've all had breakthrough drugs coming through that, and it makes a difference. It makes a difference in terms of the interaction with the FDA, the speed at which drugs get reviewed and approved, and the kinds of questions you can ask along the way. So that's been great. I think Scott, who I also have just had the pleasure to know for a long time, is actually doing a tremendous job as commissioner. It's harder to see from the outside, but he's shaking things up within the FDA. He's asking a lot of questions about why do we do things this way or that way? How can we do things faster? Obviously, the oncology division is sort of the paradigm for doing things faster, and I think he's trying to spread that across other divisions, which is going to be really good. My fear in the FDA is it's dependent upon so few people, and they're generally at the top. It's in some cases the commissioner. It's Janet Woodcock, who's been revolutionary there for 20 years and who's going to retire soon. It's Rick Pazder, who's been revolutionary there for 20 years and is going to retire soon. And I think the big question, and David and I were talking about this, is who is that next generation of leaders at the FDA and how are they going to be responding? At the end of the day, it's how they respond that really makes a difference in terms of how drugs get approved.
Yeah, I would agree. I mean, I think we've been fortunate in the oncology division that under Rick Pazder's guidance over the past 15, 20 years has really been enlightened in trying to keep up with the science. I think we've been very fortunate. Our first drug that just got approved in August was approved for acute myelogenous leukemia, as Laurie said, and it was 3 years and 11 months from the first patient who was dosed, and we got a full regulatory approval based on the first-in-human phase 1/2 clinical trial. And that's because if you're, as a sponsor, willing to partner with the FDA, and I see Ken here, and Ken and I did this 13 years ago with the development of Velcade. If you go to the FDA, at least in oncology, and say, we want to partner with you to figure out how do we get this drug approved as quickly as we can, as efficiently and safely as we can for patients who need it, they're willing to partner with you. If you come in and try and make them the enemy, then you tend to lose. So I think the real question is, as the FDA goes forward, I agree with Jeff's concern. When Janet and Rick retire, what happens? And how do we get that sentiment of creating partnerships to spill out across the other divisions as the biology is exploding in so many different fields? Jay wears a lot of hats now because he's got every therapeutic area on the planet under his domain.
Yeah. Novartis is an international company, and our corporate headquarters are in Switzerland. Our research headquarters are here in Cambridge for good reason. This is such a unique environment to do discovery and development research. But the international footprint allows you to make interesting comparisons between regulatory agencies, and working with the EMA and working with the CFDA in China. We are often reminded how forward-thinking the US FDA really is, and how much, getting to know Robert Califf, I haven't had a chance to meet Scott Gottlieb, how much that organization does with so little. The barriers to recruiting talent at every level for a government agency, and the checks required before you can get in the door, the expertise you're not allowed to have to hold a position there, which would probably help you if you did. It's amazing what they're able to do. Breakthrough status, as you asked, is a game changer. We have 15 medicines registered for breakthrough status. We use that pathway because it is so collaborative with the regulators, and we get guidance early on. It's not a commitment, but we get guidance early on as to what we both believe really moves the needle for patients. So I think the FDA has been a great partner. We could probably, we're a little conservative, our Swiss company, we could probably engage even earlier and more effectively with many of the stakeholders in this room to start a dialogue at an earlier moment in discovery science. That I'm sure we could do. The one place where I think the FDA has a huge opportunity are around the powerful incentives that they create. The pediatric voucher system has had a dramatic impact on our organization. It doesn't affect our willingness to do a pediatric study, but it does move the pediatric study much more to the front, loaded, first blush of clinical investigation. It's a powerful influence. What if such a voucher existed to develop medicines for antimicrobial resistance? On our stem cell transplant leukemia wards, we have patients with highly resistant bacterial infections. We have 4 drugs at Novartis I would love to bring in to that space. But by market calculations and ENPVs, we can't justify the spend against the opportunity presented by other more remunerative medicines. There are opportunities like...
That's an opportunity for
huge
biotech
opportunity.
and about opportunity for academia.
Correct.
Right.
So I think there's a lot of opportunity for collaboration, and I think that the incentive systems in place and that could be created are very powerful levers to drive medicine in the direction society wants it.
I think this has been a terrific panel, and let's thank our 3 wonderful panelists.